Hi guys, let's continue with our practice sessions. Okay, so the paper that we are discussing today is NEET 2018 paper. So we have taken a few questions from there and we are continuing with the discussion. So the first question that we have over here is the correct order of steps in polymerase chain reaction or PCR is. So what do we have here? Now this is the question from the chapter Biotechnology Principles and Processes. Okay, so here we look at PCR, that is polymerase chain reaction. Now polymerase chain reaction is a process of gene amplification. Okay, now what do you mean by gene amplification? See, for example, let's imagine that you're a forensic scientist, yeah, and you are collecting some data from the crime scene, or for that matter, you are carrying out rDNA technology, and the DNA that you have, the amount of DNA that you have is pretty less. So that amount of DNA is not enough for you to carry out the experimental procedures. So you need more amount of DNA. Now what is going to happen if you are going to try and clone that DNA through living cells such as E. coli or some animal cells? The problem over here is that it will take some time, okay? And with experimental procedures, you understand that time is of prime importance. So PCR or polymerase chain reaction is a gene amplification technology that gives you multiple copies of the DNA molecule, the gene segment, in short amount of time. Okay, so it has a few steps, and those steps are what? Those steps are denaturation, annealing, and extension. Okay, so when we talk about denaturation, annealing, and extension, let us understand that if this is the DNA molecule, 3' to 5' and 5' to 3' DNA structure, what is going to happen over here is this DNA structure needs to be replicated, isn't it? This DNA structure needs to be replicated, and this replication is going to require certain enzymes, certain processes, right? And this process majorly works on the principle of DNA synthesis, DNA replication. So the first step is denaturation, where the double-stranded DNA is converted to single-stranded form. Yep. So denaturation is where hydrogen bonds between the complementary bases are broken so that the double-stranded DNA is converted to single-stranded form. So you have two pieces of DNA, two strands of DNA. Now the next step is the annealing of primers. Now remember that the enzyme that is being utilized is a polymerase. Okay, polymerase, which polymerase we utilize over here? We utilize the enzyme Taq polymerase. Taq polymerase comes from the organism Thermus aquaticus, okay, which is a thermophilic bacterium. So Thermus aquaticus is a thermophilic bacterium. That means it is found in hot sulfur springs or areas where, you know, there's the temperatures are very high. So it's Thermus aquaticus. Its DNA polymerase, Taq polymerase, is stable at high temperatures. So denaturation takes place at about 92 degrees Celsius. Annealing takes place at about 42 degrees Celsius, and extension takes place anywhere between 72 to 75 degrees Celsius. Yep. So annealing of the primers means what? So if these are the primers are going to get attached to the 3' ends on both the single-stranded DNA forms, and then Taq polymerase is going to take DNTPs, that is deoxyribonucleotide triphosphates, and add them beyond this or after the primers and extend, and what you're going to get is the complete DNA molecule. So now what has happened over here is you had one DNA molecule, it has got replicated into two DNA molecules, so on and so forth. So denaturation, annealing, and extension takes place one after the other, and it constitutes one cycle of PCR. Okay, so this is one cycle of PCR. And this occurs for about 3 to 5 minutes. So one cycle of PCR takes about 3 to 5 minutes, right? And PCR, or polymerase chain reaction, is carried out for about 28 to 30 cycles, because beyond that the reliability of PCR decreases. Okay, so denaturation, annealing, and extension is also known as polymerization, by the way. Okay, so if you come across this term, do not get confused. So extension is also known as what? Polymerization. So this is the sequence of steps in PCR, and that is how we get DNA molecule. Remember that PCR is responsible for exponential amplification of DNA. Okay, when we talk about exponential amplification, you can use the formula 2 raised to n, where n is the number of cycles. Yeah, remember that. So this would take, so if you are carrying out PCR for, let's say, 30 cycles, yep, then 2 raised to 30, you can do the math and find out how many DNA molecules you have. Okay, so that was question number 1. Any doubt with PCR? Not yet. A woman has an X-linked condition on one of her X chromosomes. This chromosome can be inherited by genetics, okay, principles of inheritance and variation, where we are taking a look at sex-linked inheritance. Yeah. Now, when we talk about sex-linked inheritance, we are talking about what? X-linked inheritance over here. Okay, yeah, X-linked inheritance is what we are taking a look at. Now understand, simple things that you need to remember is, you all have studied dominant and recessive traits, isn't it? Yep, dominant and recessive traits you have already studied. Remember that the X-linked disorders that are present are all recessive. Yep, so these X-linked disorders, since they are recessive, there will be certain understanding to it, there is certain terminologies to it. So there's a simple table. So if I take this as the normal allele, normal functional allele, and this is the allele for the disease, yeah, then I can make a quick table that is applicable for all X-linked disorders. Yep, so we have the sex over here, male, female, sex chromosomes are going to be what? XY, XX, normal, carrier, and diseased. Yep, normal, carrier, disease conditions. So normal, carrier, there's no carrier when it comes to the male, but female can be a carrier. Diseased condition over here is X small a y, and disease condition over here is this thing, for the female. Now why is that the case? See, the female has double dose of X chromosome. Now since the female has double dose of X chromosome, if there is one defective X chromosome, the other normal X chromosome, the other normal functioning gene, the allele, can take over for the functioning of the defective allele. Okay, so in that case, the female has to be homozygous recessive, right? Because there are two X chromosomes, and that functionality can be managed. But in the male, there is only one X chromosome. The other sex chromosome is Y. So even if there is one, so if this particular single X chromosome shows us the defective allele, then the male is going to suffer from the disorder. But when we say that the male is suffering from the disorder, that means we are taking a look at the phenotype. Yep, understand the question over here. We are taking a look at what? We are taking a look at the phenotype. So phenotypic expression is possible only in male if the defective allele is with the mother. Are you getting it? Because From the mother, the defective allele may go to the son. It is crisscross inheritance. That it sex-linked inheritance is crisscross inheritance. That is, it goes from mother to son to the granddaughter, right? Or from father to the daughter and then to the grandson. That's basically how it works. So let's take a woman has an X-linked condition on one of her chromosomes. So that means what? She's a carrier. This chromosome can be inherited by. Okay, so this is the condition. Yeah, so let's do a quick Punnett square. Yeah, let's take it that the male is normal. So XA normal condition, X small a, XAY and XAY. So can you see that the inheritance can be seen in both the cases, but phenotypic expression of the disease will be seen only in the male. The female will be phenotypically normal but genotypically carrier. Got it? So in this case, the answer becomes both sons and daughters. Understood this? Simple enough? Calcium is important. Next question. Calcium is important in skeletal muscle contraction because it binds to troponin molecule to remove the masking of active sites on actin for myosin, activates the myosin ATPase by binding to it, detaches the myosin head from the actin filament, prevents the formation of bonds between the myosin cross bridges and the actin filament. Now this brings us to something which is called as the sliding filament theory. Yeah, or a theory of muscle contraction from the chapter locomotion and movement. Okay, so this is where you'll see this. Now, when we talk about the muscle proteins, there are two proteins, actin and myosin. Okay, the muscle proteins are two, actin and myosin. So what happens over here is, this is actin, actin, actin. Just a quick line diagram for the muscle proteins. So these are actin filaments. Okay, and then in between them what we have are the myosin filaments. Okay, what we have is myosin. Correct? We have myosin. Now, myosin is supposed to bind to actin. Okay, like this, and it's supposed to pull the actin in the inward direction. Okay, so what will happen? The actin filaments will be pulled towards the inside from both the sides, and ultimately what is going to happen over here? The muscle will contract and the action will be generated. The muscle will contract. Now, for this to happen, there should be binding between myosin and actin. Okay, there should be binding between what? Myosin and actin. If we look at the structure of actin, actin has globular actins, okay, which are arranged in the filamentous form. Okay, which are arranged in a filamentous form, that is F actin. So we have G actin globular forming F actin. Okay, now on this we have another filamentous structure called tropomyosin. So tropomyosin is present over here, and then we have some other protein molecules which are troponin molecules. Okay, troponin has 3 subunits. Okay, so that is troponin. Now troponin is a molecule that has affinity to both the actin and calcium. Okay, it has affinity to both actin and calcium. Now when it is bound to actin, the F actin, it is masking the myosin binding site, okay, on the F actin. It is masking what? It is masking the myosin binding site. So this particular point of contact between actin and myosin is what is bound, it is what is masked by troponin. So when the muscle receives a signal for contraction, what happens is a huge influx of calcium takes place into the sarcoplasm from the sarcoplasmic reticulum. Sarcoplasm is a muscle. Okay, this is the sarcomere. So Z line, I band, A band, H zone, again A band and I band. So this is a sarcomere. Yep. I band is where there's only one, there's only actin. Then actin and myosin overlapping is A band. I band isotropic, A anisotropic. H is Henson's zone. So when calcium influx takes place into the sarcomere, okay, sarcoplasms, I'm sorry, the calcium goes and binds to the troponin molecule. And because of the binding of calcium to the troponin molecule falls off from the F actin and the myosin binding sites are exposed. So now myosin utilizes ATP to form a cross bridge between myosin. Myosin's head has ATPase machinery. So ATPase is the enzyme that brings about the hydrolysis of ATP. So it brings about the hydrolysis of ATP, breakdown of ATP. The energy that is generated is utilized for the cross bridge formation between the myosin and the actin filament, and then the myosin uses another ATP to contract and pulls the actin molecules together for muscle contraction. So excuse me, the answer over here is it binds to troponin to remove the masking of active sites on actin for myosin. Got it? So that's your sliding filament theory in short. Next question. Double fertilization is fusion of 2 male gametes of a pollen tube with 2 different eggs, fusion of 1 male gamete with 2 polar nuclei, fusion of 2 male gametes with 1 egg, syngamy and triple fusion. The answer over here is syngamy and triple fusion. Okay, what is syngamy and what is triple fusion? Let's just do a quick revision of the process. Now we have the pollen grain. Okay, the pollen grain has a generative nucleus and a tube nucleus. Okay, and it has the exine, which shows us the germ pores. And from one of the germ pores, what is going to happen? The pollen tube is going to come down. Pollen tube is going to come out. Now as the pollen tube is forming, this particular nucleus, which is the tube nucleus, it moves at the tip, and this generative cell divides mitotically and forms 2 male gametes. Yep. What does it form? It forms 2 male gametes. So this is a fully developed male gametophyte. Now this development is pollen grain is haploid. Now please remember that. So the male gametes are also haploid. They've undergone mitosis. Generative nucleus was formed by meiosis. Okay. Now as this process is happening on the stigma. So the pollen tube is forming, it's moving through the style and going to the ovule. Got it? Now when it comes to the ovule, now inside the ovule we had the functional megaspore. Okay, the functional megaspore had one nucleus, okay, which underwent 3 consecutive free nuclear divisions, and with with every nuclear division it started, the embryo sac started expanding in size. Okay, so by the 3rd division there are 8 nuclei in the embryo sac. So there are 4 nuclei at each of the poles. While the cellular arrangement is taking place, yep, one nucleus from each of the pole migrates to the center. Okay, so here we have antipodal cells, yep, this is the chalazial end, and here is the micropilar end. Yep, those are the antipodal cells. The nuclei which have migrated to the center are the polar nuclei. They form the secondary cell. And over here what we have is the egg cell. And then two more cells which are synergids. Okay, yeah, two more cells which are synergids. Got it? Now, when the pollen tube enters the ovule, it ruptures at the synergids. The synergids guide the pollen tube. Okay, it ruptures at the synergid. Now when the rupturing takes place, two male gametes, the two male gametes are released into the embryo sac. This is the female gamete or the embryo sac. Yeah. So the two male gametes are released where? The two male gametes are released in the this thing, what do you call? The embryo sac. Okay. Now as they are released into the embryo sac, what happens over here is one male gamete fuses with the egg cell and undergoes fertilization. That is first fertilization, which is known as syngamy. Okay. The second male gamete fuses with this particular component. But just before the second male gamete fuses, the two polar nuclei, they fuse together and form the secondary nucleus. Yep. And ultimately this one fuses with the secondary nucleus. So here we had n plus n giving me 2n, diploid condition, haploid male gamete, haploid female gamete. In the second condition, we had haploid male gamete and diploid secondary cell, two haploid polar nuclei, isn't it? Giving us a triploid structure. So since there were three nuclei fusing, this is known as triple fusion. And triple fusion gives rise to something which is called as a primary endosperm nucleus, which will develop into endosperm, and that endosperm will nourish the developing embryo. Okay, that endosperm will nourish what? The developing embryo. Got it? This is how it works. Chalo, let's go to the next question then. Any doubts with this? Double fertilization? No?
No, sir.
Great, Raghav. All of the following are part of an operon except, okay, this is a question from molecular basis of inheritance. Okay, so let us understand what is an operon. An operon is a cluster of genes, structural genes with, I'll just write structural genes with regulatory elements. Yeah, structural genes with regulatory elements. Now, what are these regulatory elements? These regulatory elements are the promoters. Now, just let's go back to central dogma first. So we have DNA. DNA undergoes transcription to form mRNA. mRNA undergoes translation to give us the protein. Now, when this is happening, what part of the DNA is undergoing transcription translation? It's the structural gene sequence. Are you following that? What is it? It's the structural gene sequence. So, operons are clusters of genes which are found in prokaryotes. And operons can be switched on or switched off depending on the requirement. Are you following that? Yep, depending on the requirement. Now, For this to happen smoothly, there are regulatory elements called promoters, regulator, terminator, okay, and an operator. Okay, so what generally happens is, so if I draw an operon, or if I draw a very simple sequence of an operon, yep, how does it look? So if this is my DNA sequence in consideration, we have a promoter over here, then we have a regulatory element over here, then we have a promoter again, operator, and then terminator. Okay, and in between this what we have are the structural gene sequence. Remember that prokaryotic systems are polycystonic. That means there will be more than one structural gene between a promoter and a terminator. So this regulator produces something which is called as a repressor protein. In prokaryotes, transcription and translation is coupled. So the RNA polymerase, when it moves on the DNA and forms the mRNA by copying the 3' to 5' strand, this is the template strand, okay, 3' to 5' strand, what happens is, the moment a small amount of mRNA is formed, ribosomes sit on it and they start translating it. So there is coupled transcription translation. So by the time the RNA polymerase is reaching this promoter and is trying to move ahead than the operator, a repressor protein is already formed. Okay, because of coupled transcription translation, the repressor protein is already formed, and this repressor protein goes and binds the operator locus. And since it goes and binds the operator locus, what actually happens over here is it creates hindrance to the RNA polymerase, because of which RNA polymerase cannot move ahead and transcription translation is not going to occur. If these so the gene products are not going to come into picture. So when the prokaryote does not require the enzymes from these genes for its biochemical processes, the operon is switched off in this particular manner. When it requires the enzymes from this particular operon for its functioning, the operon will be switched on. That is, the repressor protein will be removed. Like for example, we have something which is called as lac operon. Okay, lac operon is lactose operon. So the enzymes that are present, they are responsible for the breakdown of lactose to be used as a source of energy. Now lactose over here is the inducer. This repressor protein has affinity for lactose also. So when lactose binds to this repressor protein, the repressor protein falls off from the operator locus and mRNA can move ahead and bring about the transcription translation, for these particular genes. So the products of these genes are going to bring about degradation of lactose, right, for energy production into glucose, galactose, and so on and so forth. And ultimately, energy will be produced. It will be utilized. And ultimately, this particular lactose will also be broken down, and this protein will be free to go and bind to the operator. And again, when lactose is not in the system, operon will be switched off. So the sequence that is not a part of the operon, we have the operator, the promoter, and the structural genes. So the enhancer is not the part of the operon. Got it? Moving ahead, match the items given in columns, column 1, column 2. Okay, wonderful. Fibrinogen, osmotic balance, defense mechanism, globulin, albumin. Okay, simple enough. Yep, you all know this. Where is fibrinogen involved? Yep. Recall that At the site of the wound, platelets arrive, and platelets release a component called thrombokinase. Yeah, thrombokinase converts prothrombin to thrombin. Yep, thrombin converts inactive fibrinogen to fibrin, and fibrin forms a meshwork, traps RBCs, and forms a clot, isn't it? It forms what? It forms the clot. It forms a blood clot which plugs the wound and prevents blood loss, isn't it? That's basically what happens over here. So fibrinogen, yes, Raghav?
Is it meshwork plus RBCs or platelets?
No, meshwork plus RBCs. RBCs are caught in the meshwork so that the clot is... platelets degenerate, Raghav.
Oh, okay.
So platelets and the epithelial cells over there, they degenerate. So platelets, when they degenerate, they release thrombokinase. Thrombokinase or thromboplastin, yep, remember the names. Globulins. Yep, globulin is responsible for defense mechanism. Why? Because globulins are antibodies. Okay, so these are actually, globulin is a family of proteins. Yep, so we have immunoglobulins, IgG, IgA, IgE, IgM, and IgJ. Okay, so these are basically the different globulins that are responsible for defense mechanism. So that leaves us with albumin for osmotic balance. Okay, osmotic balance is what? The water balance of the blood. So the answer A2B3C1, right? Next, another one. Oh, this is menstrual cycle. So human reproduction. This particular, the previous question was a combination question, by the way. Since your fibrinogen and what do you call that? Blood clotting comes from circulatory system from 11th grade, body fluids and circulation. Globulin will come from human health and diseases from 12th grade, okay? And albumin will basically come from where? If it's osmotic balance, it's coming from excretory products and their elimination. Human reproduction, this is menstrual cycle. So menstrual cycle, simply we are discussing phases. So the menstrual phase or the phase of menstruation. Then we have what? Follicular phase. Then the ovulatory phase. And finally, the luteal phase. Yeah? What do we have is the luteal phase. So menstruation is the first phase that occurs from the 1st to the 5th day of the menstrual cycle. Follicular from 6th to 13th day of the menstrual cycle, ovulatory on an average on the 14th day, and luteal phase from the 15th to the 28th day of a menstrual cycle. So a 28th day cycle is going to give us this, right? So menstruation shows us bleeding wherein the endometrial lining of the uterus is shed, it's broken down, and it comes out through the vagina in the form of blood and mucus. Follicular phase is where the follicles are growing, okay? Then they're moving from primordial follicles to primary follicles to secondary follicles, that is early and late secondary follicle, and then to the Graafian follicle. And while they are doing this, they are releasing estrogen, which is rebuilding the endometrial lining of the uterus. So it is proliferative. Okay, it is what? It is proliferative, right? Ovulatory is where ovulation is taking place, where the Graafian follicle is rupturing and the secondary oocyte, the ovum, is being released into the fallopian tubes. Luteal phase is the secretory phase. Why? Because it is responsible for secretion of progesterone, which is maintaining the lining of the endometrium in anticipation of pregnancy, and it's the pregnancy hormone. Got it? Yeah. So A2, B3, C1. Next question. Secondary xylem, okay, and phloem in dicot stem are produced by. Let's understand this. So when we talk about xylem and phloem, what are we talking about? We are talking about vascular bundles, isn't it? Yeah. There are plenty of hints over here. Secondary xylem and phloem. The term secondary over here tells us that we are talking about secondary growth, which is a characteristic of what? Dicot plants only. Now, why is it the characteristic of dicot plants? What type of meristem is responsible for secondary growth? Lateral meristem, which is also known as cambium. So what is the only option that tells you the answer? Vascular cambium, isn't it? Yeah. So hints are in the question itself, but let's understand this. So when we talk about the dicot stem, in the dicot stem, the vasculature, okay, is called open type. Okay. What do you mean by open type? Open type means it shows secondary growth. Okay. It shows what? Secondary growth. Remember that phloem is always towards the outer side and xylem is towards the central side. So center side is the pith. Okay. And here you will have what? The epidermis. So epidermis is the outer side, right? So we have the phloem over here and we have the xylem over here. So this is the phloem, this is the xylem. And in between the xylem and phloem, what we have is the vascular cambium. Are you getting it? It's the vascular cambium. And this vascular cambium is going to give rise to cells in this particular direction and also in this particular direction, forming secondary phloem and secondary xylem. So now another term. So please don't get confused if you come across these terms. See, xylem and phloem is a vascular bundle. A bundle is a fascicle. Yep. So this cambium is also known as intrafascicular cambium. Okay. And then there will be more phloem xylem like this. And this particular cambium, which will be present between two vascular bundle, will be known as interfascicular cambium. Both of these cambia play an important role in secondary growth of the plant. Which of these statements is incorrect? Oh, respiration. Enzymes of TCA cycle are present in the mitochondrial matrix. Glycolysis occurs in cytosol. Glycolysis occurs as long as it is supplied with NAD that can pick up hydrogen atoms. Oxidative phosphorylation takes place in outer mitochondrial membrane. Great. So let's recall the process. So we have glucose. Glucose undergoes glycolysis. Where? In the cytoplasm. And forms what? Pyruvate. Now this pyruvate, if it stays in the cytoplasm, it is going to undergo anaerobic respiration. And depending on the organism we are dealing with, it might form lactic acid if it is human muscle cells. It might form ethanol if it is plant cells. But pyruvate now undergoes link reaction in the cytoplasm itself and forms acetyl-CoA. This acetyl-CoA moves into the mitochondria and undergoes tricarboxylic acid cycle, or the TCA cycle, or the citric acid cycle, or Krebs cycle. All names, look out for them. And ultimately undergoes aerobic respiration. After TCA, there is oxidative phosphorylation, which is also called as terminal oxidation. Yeah. So this entire process will be aerobic respiration. Get it? Now TCA is occurring in the inner mitochondrial matrix. Okay, I mean matrix of the mitochondria. So enzymes of TCA are present in the mitochondrial matrix is correct. Glycolysis occurs in the cytosol. Yep, is correct. Glycolysis operates as long as it is supplied with NAD that can pick up hydrogen atoms. Yes, in the second phase of glycolysis, the payoff phase, there are oxidative steps where the protons are removed from the substrate and NAD picks up those protons to become NADH. This happens in 2 steps in glycolysis. Okay, so that is also correct. Oxidative phosphorylation takes place in the outer mitochondrial membrane. That is wrong, because oxidative phosphorylation takes place in the inner mitochondrial membrane. The inner mitochondrial membrane has certain components called as coenzymes and electron carriers. Okay, and these coenzymes and electron carriers, so they basically pass on the proton, the electron from one to the other, and the ultimate electron acceptor is oxygen. That is the reason it is known as terminal oxidation. Okay, are you getting it? And since these are electron carriers, they will be passing on electrons further. And this happens on the inner mitochondrial membrane. And because of electron carriers, oxidative phosphorylation or terminal oxidation is also the electron transport chain. So the incorrect option is oxidative phosphorylation takes place in the outer mitochondrial membrane. That is wrong. It occurs in the inner mitochondrial membrane. Next question. Which of the following terms describe human dentition? Simple enough. Yep. Our teeth are embedded in the jaw, right? The jaw is the part of the skull. Yep. Don't, yeah, is related to dentition, teeth. Okay. Now, a theca is a lobe or a cavity. Okay. So, thecodont means what? It is embedded in the jaw. So, thecodont is correct. So, now out of these options, there are only 2 options that show us thecodont, that is A and B. So, let us find out the correct one from there. Diphyodont. Diphyodont means that the teeth appear in 2 stages. Is that the case with the humans? It is, isn't it? We initially get milk teeth, which are replaced by permanent teeth later, isn't it? So, diphyodont. And then Are all of our teeth the same? No, no. We have incisors, we have canines, we have premolars, and then we have molars. Incisors are for biting, canines are for tearing, premolars and molars are for chewing and grinding, isn't it? Yeah. Then, so since the teeth, the dentition, the teeth that we have are different, the condition is called as what? Heterodont, right? So, the option B, that is thecodont, diphyodont, heterodont, describes human dentition. This is from the ecology unit from your 12th grade. What type of ecological pyramid would be obtained with the following data? Secondary consumer, primary producer. Okay. One thing that you always need to remember is that the pyramid of energy can never be inverted. So this is an upright pyramid and this is an inverted pyramid. The pyramid of number and pyramid of biomass can be inverted, but the pyramid of energy will never be inverted. So pyramid of energy is gone out of it. Yep. So when we talk about pyramids, generally, what do we talk about? We talk about the base is the organism which is high in number, isn't it? Yep. So we generally have producers over here, then the primary consumers, and then the secondary consumers. Yep. Now that is dependent on the number and the biomass. So it says, what does it say? It tells us whatever ecological pyramid will be obtained with the help of the following data. 120 grams. So if it is grams, then we are talking about biomass, isn't it? So the primary producer is 10 grams. Second primary consumer is 60 grams and secondary consumer is 120 grams. So my pyramid over here is inverted, isn't it? Because the producer, which is supposed to be at the bottom, is less. 10, 60 and 120. Yep. So this will be an inverted pyramid of numbers, and pyramid of biomass can be inverted, but pyramid of energy can never be inverted. Remember.
Is this 12th grade portion?
Yeah, ecology. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Okay. Simple enough. TI plasmid is a tumor-inducing plasmid which is found in an organism called Agrobacterium tumefaciens. Agrobacterium tumefaciens is the organism which is utilized for gene transfer mechanisms in dicot plants. Okay. So with the tumor-inducing plasmid has tDNA, and this tDNA forms something which are called as crown gall tumors. Crown gall tumors are harmless for the plants. Okay. So if there is a crown gall tumor, that means the tDNA has been introduced into the genome of the plant. Okay. So what is done? The gene of interest is linked to the tDNA. So the tDNA, as it incorporates itself into the plant genome, it also incorporates the gene of interest in the plant genome. Okay, so some beneficial gene that you want for the plant, you can introduce it with the tDNA. So because the tDNA is linked with the gene of interest, and if we see the crown gall tumor in the plant, that means the tDNA has got incorporated. And if the tDNA has got incorporated, that means the gene that we had linked with the tDNA has also got incorporated. So tDNA has worked as a marker gene for confirmation of successful transformation. Lambda-phage. Lambda-phage is a bacteriophage. Okay. Bacteriophages are the ones that infect bacteria. Yep, and they multiply inside the bacteria, bring about the lysis of the bacterial cell, and they spread. PBr322 is a plasmid which is utilized for recombinant DNA technology mechanisms for transformation of bacterial cells. Yep. Retroviruses, on the other hand, are viruses which are specific to mammalian cells, mammalian hosts. Okay, so like HIV, all of these, HIV, etc., these are retroviruses, so they can infect the mammalian cells. So which of the following commonly used vector is used for introducing DNA fragment in human lymphocytes? Then it's going to be retrovirus. A classic example of this is gene therapy for adenosine deaminase deficiency. Yep, adenosine deaminase is an enzyme that is required for the proper functioning of T lymphocytes. Okay, so deficiency of adenosine deaminase causes a defective immune system. So in this gene therapy, what is done? A functional ADA gene, adenosine deaminase gene, is incorporated into the retrovirus, and this retrovirus is allowed to infect the T lymphocytes of the person, of the baby rather, that is suffering from ADA deficiency. And these lymphocytes, which now carry the functional ADA gene, are put back into that baby who is suffering from adenosine deaminase. Adenosine deaminase deficiency is known as severe combined immunodeficiency disorder. SCID, SCID. Got it? Simple enough. So, retroviruses. Let's move ahead with the next question. What is the role of NAD plus in cellular respiration? Okay, let's understand two things. There are two components, NAD and NADP, that is nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, both of which act as proton acceptors. Yep. So these can form NADH, this can form NADPH. Always remember that NADH is seen in catabolic reactions and NADPH will be seen in anabolic reactions, that is building up reactions. So NAD will always be seen in respiration and NADPH you'll see in photosynthesis reaction. So don't get confused between the two, where to put NAD and where to put NADP. If it is photosynthesis, it is NADP and NADPH. If it is respiration, it's NAD and NADH. Got it? Please remember the differentiation. What is the role of NAD plus in cellular respiration? Yep, it functions as enzyme. No. Okay, it basically functions as an electron carrier. We were just discussing oxidative phosphorylation or terminal oxidation or electron transport chain. So NAD is one of the electron carriers that is present on the inner mitochondrial membrane. The electron transport chain begins with NAD and ends with oxygen. Okay, this arrangement is based on what? Based on the energy of these particular components. So NAD is a very high energy compound, oxygen is the least energy compound. So from high energy to low energy, the arrangement of electron carriers is on the inner mitochondrial membrane. Got it? Which of the following is not a product of light reaction or photosynthesis? Yep. Now come on, I literally gave you the answer just on the previous slide. Yep. Light reaction or photosynthesis has cyclic photophosphorylation, and non-cyclic photophosphorylation. Cyclic photophosphorylation happens to increase the number of ATPs that are produced. Non-cyclic photophosphorylation gives us ATP plus NADPH plus O2, right? Yep. So O2 is a product of photolysis of water. Yep. ATP is photophosphorylation, NADPH is in non-cyclic photophosphorylation, the electrons that come out from PS1. ATP in cyclic photophosphorylation. Got it? So the answer over here is NADH. Remember that ATP and NADPH are together called as the assimilatory power. Okay, and it is utilized in the Calvin cycle for glucose synthesis. Yeah, got it? So a total of 12 NADPH and 18 ATP are required for the synthesis of one glucose molecule through the Calvin cycle. Which of the following is an amino acid derived hormone? Now this is from your paper, so understand. This is very easy to understand. Always look at the endings. Okay, when it is INE, it's an amino acid derived hormone. If it is ONE, as in case of testosterone, progesterone, or iol, as in case of estradiol over here, or estriol. Estradiol is estrogen. Okay, this will be steroid hormone. Got it? So the answer, you can definitely tell me right now that it is epinephrine. Epinephrine is obtained from an amino acid called tyrosine. Got it? So remember this bifurcation. INE is amino acid derived. O, if it ends in ONE, and IOL is a steroid hormone, and the source, the parent molecule, is cholesterol. Got it? Among the following sets of examples for divergent evolution, select the incorrect option. Divergent evolution leads to the formation of homologous organs. Yeah? Homologous organs are the organs which have differentiated from a common ancestor. Okay, so look at the options over here that tell you a common ancestor. Man, bat, and cheetah, all of these are chordates, vertebrates, so obviously they have a common ancestor. Yep. Among the following sets of examples for divergent evolution, select the incorrect option. So obviously we have to select the incorrect option over, so obviously option A is not there. Heart of bat, man, and cheetah. Again, bat, man, and cheetah, same ancestor. They're diverging from the same ancestor. So again, this is an incorrect this is not the answer over here. Brain of bat, man, and cheetah. Doesn't matter what organ we are talking about. There's a common ancestor in all of these three options. So obviously these options are not available over here. These options are not applicable over here, sorry. Eye of octopus, bat, and man. Octopus is an invertebrate, isn't it? So the ancestor is different. Got it? So the answer over here simply becomes, for divergent evolution to form homologous organs, there should be a common ancestor. And the last question that we have is, offsets are produced by meiotic divisions, mitotic divisions, parthenocarpy, parthenogenesis. Remember that offsets are vegetative propagation, isn't it? And vegetative propagation occurs through the vegetative parts of the plant, that is root, stem, leaves, etc. If it is the flower, then it will be sexual reproduction where meiosis will be taking place. If it is vegetative propagation, it's direct mitosis. Got it? So more cells will be multiplying by mitotic divisions and offsets will be formed. So this was with a set of questions for today. Yeah, I shall be taking a few more questions from another set of papers, previous papers, and we'll be continuing with the concepts. Yeah. Bye guys. Have a nice day.